to identify the viruses and what is their multiplication, comparing viruses to living organisms, the smallest one being bacteria, and so on. So in this chapter 13, we have two parts. The first part is in chapter 13, supplement. There you have the questions. After few concepts, you have questions that ask you about those concepts. So it's like you use those questions to review what you just covered. And that will go up to the taxonomy of bacteria. That will be the end there, okay? So, and then you take the chapter 13, just the 13. Don't use the first part because you see multiplication, all that you have already in supplement. But in chapter 13 period, you start with taxonomy of viruses. So in that you have the taxonomy, how to classify and name the viruses. 2, how to culture viruses in the lab. That's where we saw that we use host cell. Without host cell, the viruses cannot multiply. So if the virus is a bacteriophage, the host cell will be the bacteria. So when we affect the host cell, what are the marks, the print of viral infection on bacteria? That is called plaque, the clearing on the lawn of bacteria. But if the virus is animal virus, you will have the print is called cytopathic effect of the virus on the host cell. Then we did talk about to culture, what do we need? For bacteriophage, you need bacteria. But for animal viruses, you can have in vivo, that means inside of living animals that can be fertilized egg or a small animal or even big. You have dogs, you have cats, you have chimpanzee, you have the mouse, so small, big, but the animal where you inject the virus and then take certain organ from that animal, study them to see the cytopathic effect of the virus on those organs. You can also do the in vivo. People doing the research often is in vivo. Instead of growing the virus in the animals, you grow them on animal cells. The question was, where do we get the animal cells? There are two sources. We can use the cells that are normal cells and the cells that are transformed, like cancer cells. What is the difference between the two? We did that last time. The normal cells have short time of life. 2-3 weeks, they die. So if you had to do your experiment, you try to do it in first and second week. The transformed cells live forever. Number 2, the normal cells have what we call encourage dependence. That means they depend on the support. They need the support to attach to, then they will divide. If there's no support, if they are not attached, they cannot divide. That is encourage dependency. While the transformed cells did lose that, they can divide even in suspension, they will keep dividing. 3, the normal cells have what is called contact inhibition. They grow in 2 dimensions, but once they touch one another, they stop multiplying. So that is contact inhibition. While transformed cells they lose the contact inhibition. That means they can touch each other, they will continue to divide. What is the result? When the two dimensions are filled, they will keep dividing in height. So you have three-dimensional structure, like tumor, okay, which you cannot have with normal cells. Finally, the normal cells produce the normal amount of growth hormone that can secure the pace of life cycle of the cell, while transformed produce a large amount of hormone, growth hormone, that will secure the cell cycle and make it go quicker and quicker in such a way that after a while you have too many cells made because the cycle is quicker. Too many cells that are made, so you have a mass of cells that do not have a special function. They are non-functional. And that mass is the one that can form the tumor. And we just say that the tumor can be benign, no cancer, or the tumor can be malignant. With cancer, you have some protein, cancer protein that will appear on those cells. And we did also say that when they are in tumor, often they have a membrane around them that confines them together. But when that membrane breaks, the cells will migrate in different parts of the body. We will talk about general cancer or metastasis. So besides the culture, we did see that how to identify the virus. Say that this is herpes virus, this is cytomegalovirus, and so on. There are three techniques for identification. You have cytopathic effect that you recognize on the host cell. Two, you have serologic procedure using the serum to see the formation of complex between virus and antibodies.